My name is Bulow Møiniche. I would like to present our recent review in Allergy titled Mapping Allergen B and T-cell epitopes, Technological Advances, and Their Role in Precision Allergy Therapy. So, allergic diseases are driven by highly specific interactions between the adaptive immune system and the allergens. And rather than recognizing entire allergens, B cells and T cells recognize discrete molecular regions known as epitopes. B cells recognize the structural features on intact allergens and produce IgE antibodies, while T cells, on the other hand, recognize allergen-derived peptides that are presented on HLA molecules after antigen processing. Importantly, increasing evidence suggests that allergic sensitization, disease severity, and also treatment outcomes are governed at the epitope level rather than at the whole allergen level. So, for B-cell epitopes, recent technological advances now allow increasingly detailed characterization of IgE recognition. Classical peptide-based approaches can identify linear epitopes, while display technologies and demutational scanning enable high-throughput screening of the antibody-antigen binding regions. At the same time, structural methods such as X-ray crystallography and cryo-EM provide an atomic-level insight into the allergen-antibody interactions. Importantly, some studies have shown that clinically relevant IgE epitopes are conformational, which also highlight the importance of preserving native allergen structure during epitope mapping. More recently, AI-driven prediction tools are also emerging as a powerful approach for computational epitope mapping and allergen modeling. On the other hand, T-cell epitope mapping addresses a complementary aspect of the allergen recognition. So following antigen processing, allergen-derived peptides are presented by HLA molecules to allergen-specific T cells. And because HLA molecules are highly polymorphic, different individuals may present different allergen peptides, contributing to a substantial heterogeneity in allergic responses. So to identify these epitopes, studies combine computational prediction, peptide screening assays, and PMHC multiple technologies, and increasingly also single-cell sequencing approaches that allow a detailed characterization of allergen-specific T-cell repertoires and functional phenotypes. So finally, epitope-level insight are now beginning to translate into clinical applications. Epitope-level resolved diagnostics may improve molecular characterization of sensitization patterns and help predict disease severity and also cross-reactivity. Functional cell-based assays such as the basophil activation test can further also connect the molecular sensitization with the real clinical reactivity. And at the therapeutic level, epitope mapping enables the rational design of hypoallergenic proteins and peptide-based immunotherapies that aim to improve both the safety and efficacy in the end. So together, advances support the development of precision medicine approaches in allergy diagnosis and therapy as well. So overall, the advances in epitope mapping technologies are transforming our understanding of allergic disease and providing a molecular framework for the next generation precision allergy therapies. I hope you enjoy the read of our review. Thank you for listening.